Hi, everybody, and welcome back to Let's Combinate, where we are mastering medical device and pharma quality little q. I'm your host, Subhi Sadeh, and I'm so glad you're back for another episode. But before we dive in, FYI, I've put a Calendly link in the show notes because I'd love to get to know you, my audience, a little better. I know there's at least a few hundred of you out there. I know you hear this. Don't be shy, and let's connect. Today, I am joined by the incredible Marta, who is the CEO of Radyus Research, one of the first 10 guests of the show. She has a one-of-a-kind background, venture capitalist turned CRO, founder, and CEO. She has a PhD in immunology and an MBA from Kellogg. Very, very high on the scale in two technical and business areas. And so she's really good at navigating the crossroads of science and business, and I'm excited for her perspective today. Today's discussion is going to focus on target product profiles, or TPPs, a critical tool in drug development and an essential piece for combination products. TPPs provide a clear roadmap for aligning clinical goals, regulatory requirements, and market strategies, keeping everyone on the same page throughout the development process. For the people who work in combination products, where drugs and devices need to work together, a well-crafted TPP makes sure that we're both aligned on the drug and device side to the needs of patients, regulators, and the market. For anyone who's ever worked on a combo product and you're working through outlining your user requirements, you really can't do it without understanding the target product profile. So stay tuned. This discussion will give you a deeper appreciation for the power of TPPs and how they can make or break the success of combo products and pharma products. Welcome, Marta.
Thanks for having me.
Yeah, I guess maybe let's level set. How would you define what a TPP is for people who aren't familiar with it?
Okay. TPP is a roadmap. You mentioned it yourself. So TPP is a document, essentially, and it can be a one-page document. And in a life cycle of a product, it's almost like step one. So a lot of times, and I work with a lot of startups, early stage technologies, academic spinouts, but I think this translates into pharma as well if they're doing their own internal discovery. The moment you put together this document called a target product profile, you have moved officially from discovery into development. So psychologically, it's a very important document that for the very first time will define how our future product will look like. And I love doing TPPs with a lot of our clients at Radyus Research today. And then you've mentioned I come from the investment world. That was one of our favorite diligence documents because, in a sense, it integrates everything that shall become a label one day. So it's a very fun challenge to today, maybe 15 years before the product is approved by the FDA, put down your hypothetical label on paper and really focus your team and yourself to say, okay, I'm going away from this technology that I have into defining what are the parameters or characteristics of my product to be. How will this be dosed? What's the frequency of dosing? How is this going to be formulated? What is my target population? We'll talk about the sections of a TPP, but to answer your question, it's a very strategic document that comes from strategic thinking of what are we building? How does it need to look like? What are the specifications? And all of the fun stuff that stems from that product development on the regulatory, manufacturing, and quality side. So I love doing TPPs very early in product development.
In terms of how the document is actually controlled, I've seen it a few different ways where, because TPP is an important section of a submission, and to your point about labeling, it's something that is like rubber stamped and approved, and it shall not change after a drug is approved unless you're going for a new indication or something like that where you have the same molecule and you're branching out. But I guess my question is, a TPP a quality document that it should be under revision control and approved, or is it something that once submitted and approved by FDA or whatever health authority, that's what locks it down officially, just like your final label post negotiation?
Once the product is approved, your TPP is effectively your label. So purpose of a TPP once the product is approved is minimized because there are other documents that are governing how you're manufacturing and using your product and what the FDA has approved it. But until the moment the FDA approves the product, a TPP is an evergreen document. And it is ever changing and uses change controls to document all of that. And my world these days is mostly preclinical. So the oversight of the FDA of a TPP document is minimal, and it mostly serves as a guide for product development. But as you submit your IND and have it approved, you do submit your target product profile as an official document as part of the application. And then it becomes an official quality document that is governed by the QMS. And so it is an evergreen guidance document up until a point. Once it's approved, then you got what you got. It's no longer a hypothetical future product, which TPP describes. It is now an approved product with particular qualities that the FDA has approved. Yeah.
In other words, it goes away from a target to just a product profile.
Exactly. Yes.
Yeah,
Yeah.
yeah.
And until it's approved, that thought process of what is that we're building and how are we using the information that's in a target product profile to improve it? Because at the end of the day, the TPP is telling us how are we going to be competitive and how are we going to manufacture a high quality product in a controllable way while making money.
Talk a little bit more about that last part.
So again, drawing inspiration from a lot of preclinical programs that I'm involved with right now under Radyus, we deal with companies that underestimate the power of a TPP. In other words, there's a common misconception that until you put a product in humans in the clinical trial, you don't really have to think about much about how are you going to compete in the clinic and define certain things. And you don't have to think about how you're going to be manufacturing and what's going to be your cost of goods sold. And you don't have to think about impurities or certain quality aspects that can drag on your manufacturing.
We'll
So
just deal with it. We'll just deal with
we'll
it
just deal
later.
with it. It's not important right now. Right now, I'm doing this mouse model study to show efficacy. Right now, I'm not focused on how this is made and how this is controlled. And I'm not really worried about tox in the clinic because I'm worried about my preclinical animal tox, right? So there's a lot of compartmentalization in preclinical product development that, if not addressed in the preclinical stage, can really negatively impact product development in the clinic, manufacturing, and quality. And so, if your quality organization gets a product that's already in the clinic, but has not thought through certain quality aspects of manufacturing early on, or process development, or critical quality attributes at the preclinical stage, the margin to change that target product profile as the asset progresses from preclinical into phase 1, phase 2, phase 3 is narrowing down. The levers that you can pull to improve that target product profile so you can be more competitive and more appealing to the FDA for the approval are minimized. So if you have a really large gap at the preclinical stage, there's only so much that manufacturing, your CDMO, can do to fix that, if that makes sense.
No, totally. And I remember I used to work in commercialization at a big medical device company at the time. And they would use this euphemism of throwing things over the wall, where, and
Yeah,
actually what's really hilarious is people who work in manufacturability have this saying of it's put together with duct tape and bubble gum.
that's
And
right.
then you're supposed to just commercialize it.
Yeah.
Yes, a topic that I've had a lot of conversations with people about, but I think okay. This is something that I find both true and frustrating at the same time. Is you talk to a business person and they tell you you need to figure out the way to, like you talked about, what is a commercial market access strategy. You
Mhm.
talk to a regulatory person and they say you got to start as soon as you figure out as soon as you initiate a project, you need to figure out regulatory strategy. You
Mhm.
talk to a quality person, like you just said, CQAs. And you can't start everything all at the same time. And you can't you can't handcuff early development by saying you have to do everything that you would do for commercial for something that's small. And while I agree on principle, it's something that you should be considering and something that you don't want to be blindsided by. In practical terms, what does that actually look like? That what is the because I think what gives me at least heartburn as a normie person who doesn't really deal with these things in detail is when I hear start as early as possible, I hear you need to do the thing that you would do post pivotal study before you put first in human, kind of like. And I
Yes.
know that's not what you're saying. So what does it actually mean to start early and have these considerations early on?
Yeah. And it is at the root of what makes a program successful versus not. And I would argue that a TPP, putting it together at the preclinical stage, when you're at the candidate selection stage, before you qualify your clinical candidate, is the best time to write a TPP because it does address the commercial competitive information. It does include manufacturing and quality section. It does include the expectations for efficacy and safety preclinically and clinically. And it forces you as a drug developer at that candidate selection stage to start thinking and putting seeds and plans for the downstream activities. It's not going to be as involved. So usually companies, if they're in the preclinical stage, they don't have robust QMS system. But by putting on paper, and it can just be 2 pages of a TPP at a candidate selection stage before you go in front of the FDA for the pre-IND meeting, so not even IND application, and saying, this is how I'm thinking about the target indication. This is my mechanism of action, and these are the biomarkers that I'm going to use. And that's going to tie into my clinical trial design, plus maybe quality release criteria for the manufacturing, how I'm going to release my release assays, right? That goes in the TPP. That type of granularity goes in the TPP, and it forces you to start thinking about, okay, what's going to be my drug formulation? Is this going to be oral? Is this going to be IV? So route of administration is going to drive the formulation. What am I expecting my stability to be based on my modality that I have? What are my purity expectations? Based on the purity expectations, this is my toxicology expectation on safety that I'm going to demonstrate preclinically and then clinically. So by following the cookie cutter sections in the TPP document, you start thinking about some of the quality aspects, the manufacturing aspects, the safety aspects that have to be addressed today. Maybe not in a 100-page document that's quality control, change control, govern, but it is a strategic document at this stage, and then it becomes a more official document. And we'll talk about, I mentioned some of the sections in the TPP. It does include quality and manufacturing and safety and efficacy and commercial differentiation element, risk mitigations, everything under one roof. At the candidate selection stage, 12 months before you even dose the first patient, and that's what separates successful programs from unsuccessful programs. So when somebody tells you, oh, this failed preclinically or whatever, chances are they didn't have a robust TPP and they were not updating it regularly.
So let's then level set on what goes into a TPP. Are there regulatory expectations for what goes in a TPP, and is there any exercise where you have to reconcile, say, FDA versus EMA expectations of what goes in a
Yeah.
TPP?
Yes, 100%. So
Where are
you're
they
ready.
defined?
So let's talk about there are different flavors of TPPs. I come from a drug world, so not the combination product or devices, but the principle is the same. A regular TPP will have 10 chapters or 10 sections. You're ready to write a TPP, which is your future label, once you know what your modality is and what is your target indication. You cannot write a TPP if you're a platform company and you don't know what your target indication is. So, out of the 10 sections that are in the
Sorry,
T,
before you move on, can you, in layman's terms, modality and target indication?
oh, sure. So, again, this is for those of us who are in the preclinical stages, moving from discovery into the development or from just pure R&D within pharma. Oftentimes, new therapeutics start with target identification. You identify a signaling pathway that modulates a certain disease. And then our goal as drug developers is to find an entity that's going to modulate that signaling pathway. It's either going to block it or activate it or you name it. And oftentimes, you have to think in terms of what's the right modality to modulate that pathway. Is it a small molecule? Is it a monoclonal antibody? Is it an antibody drug conjugate? Is it cell therapy? Is it something different, gene therapy? So that's what I think of when I say modality. Oftentimes, there is very robust information on the biology of the disease with the new target, but a wrong modality is chosen. They maybe develop a monoclonal that doesn't penetrate inside a cell effectively and it can get engage an intracellular target. And then maybe a small molecule might be a better way to address it. And a lot of times we see a mismatch between a modality and the druggable target. So if you have that mismatch, that's going to drive your dosing. So now you'll have to dose twice or three times more just to reach your therapeutic index, right? So choosing the right modality has to be done early on. So that's effectively what you drug. Is it a small molecule? Is it a monoclonal? What is it? What's that product? And that modality is going to drive the specificity, the dosing, the tox profile, everything that you are thinking about on the QA side in the clinic or down the road. It's driven by the choice of the modality. If it's wrong, you're dead in the water from day one, and you could try to do as much as you can that later on, but you may just fail. So that's what I mean. And you have to have that modality locked in, and you have to know for what indication you're developing a therapeutic.
Does whether it's like IV or sub Q or solid oral dose, does that play into? Because I guess my question is, let's say let's use small molecule as an example. You can have a small molecule pill, you can have a small molecule sub Q injection, you have a sub Q molecule that's compounded into a bag, and
Yeah.
then large volume IV, intramuscular, so on. So
Yeah.
does that play in, is that included in modality, or is it just the framing of, is it a mAb or a gene therapy?
That's included in the TPP. That is,
Is that, does that included in the is that modality?
yes, that is basically in a modality because that's going to be driving how the drug is used and what the label is, right? And so the right modality has to be chosen with all of those things in mind.
Okay.
And
So modality basically frames out like is it, this is probably like two layman's terms, but is target indication what and modality how?
the other way around. The modality is what? What are you making? What's the physical product that you're making? And then the indication is how are you using that product?
Okay. Okay. No, that makes sense. And
Yeah.
then, okay, so target indication, how much detail is in a target indication?
Quite a lot of detail because you will need to follow the FDA guidelines for that particular indication. So that's why to start doing a target product profile, you have to know the indication and the modality because the FDA issues guidances for the development of modalities separate of the indication. So now you have your indication selected. What is going to be the inclusion criteria into my clinical study? Am I going to do any biomarkers to select the patient population? Is it like pediatric? So you have to start thinking about that so that you can pull down the right FDA guidance for that one. And then you pull down the FDA guidance for the modality, let's say small molecule in oncology. And then you read both. And then you start thinking, okay, the FDA wants me to do these preclinical studies. And this is how they want me to think about clinical development. That you need to know before you select your candidate, before you start writing your TPP.
Uh-huh.
So in
And go on.
Yeah, so indication, target indication is very critical. That's where you're like, okay, how are these patients treated today? Because what is a target product profile? It helps you understand how you're differentiated. That's one of the 10 sections of the TPP is the differentiation. So once I select my target indication, let's say it's going to be ALL in a particular demographic of patients, and it's going to be refractory ALL. So they have failed other therapies. So that's going to
What
be
is ALL?
the leukemia. Oh, shoot, I'm blanking on it. It's a form of a leukemia. So you will define what those patients.
Acute lymphoblastic leukemia
There you go.
is a type
Thank you.
of blood cancer that occurs when bone marrow produces too many immature white blood cells called lymphocytes.
Great. Thank you.
Okay,
So
you got it.
you will start your TPP by defining whom you're treating. So you have your modality, and then you say, okay, I'm going to be treating ALL patients that fit this criteria. Well, I'm not going to be first line. I'm going to be second line. They're going to be refractory. They're going to be very sick patients, right? This is going
What
to
does
be the
refractory mean? Sorry.
refractory means they have failed other previous therapies. So they're
I
not
see.
responsive to other therapies. So that's the connection to the label, right? If you think in terms of what's on the label when you get a new medicine and then you pull up the label, that's basically the first paragraph is like, this product is indicated for this particular population of patients. That's what they got the
Mm.
FDA approval for. So now we're like, traveling into future 15 years from now, what's going to be on that label? That's the very first chapter of your TPP is let's pretend that we can compete in the ALL population that's like this. And this is how many patients there are and all of that fun stuff. That's going to drive like, why are you developing this product in the first place? Right? A lot of times when there's no TPP written early on, there's no clarity on how this compound is going to be used and what they're going to be treating. And so you have to put yourself in the shoes that you already have this product in hand. The FDA has already approved it. How are we going to compete? What are we expecting to get out of this one in terms of the efficacy now? So that's effectively the second chapter or second paragraph of your TPP is, okay, now we've selected our target indication. What efficacy thresholds are we expecting in the clinic? So we need to look forward and say, do we want 60% reduction in tumor size, or do we want overall survival rate to be our endpoint? And then we need to find out who else is treating those patients in that indication so that we know what good looks like. And that's where the target in the target product profile comes in. You have to define the efficacy threshold and how are you going to be measuring that preclinically in the relevant animal model and clinically.
In terms of aligning between different regulatory bodies, is there any differences between, say, FDA or EMA?
Yeah, there are. And one of those sections of a TPP is a regulatory strategy that you're touching on. Maybe even high level, is this going to be an NDA or is this going to be a 505(b)(2, right? Maybe you're taking and repurposing a drug. You have to know what the regulatory pathway is and what your regulatory body is expecting you to show preclinically and clinic. And so you have to know the ICH requirements or at least inform yourself. We talked about the evergreen nature of a TPP. Do I really need to know it today, or maybe in 3 months I'll add it on? That's going to grow. But yet you will need to decide in your section of a TPP that says regulatory strategy, what are the geographies that I'm going to be commercializing this in? Is it US? Is it China? Is it Australia? Is it Europe? Which countries in Europe? So what do I need to demonstrate? What's the regulatory path? Can I do accelerated approvals of any kind? How do I do that? That goes in a TPP. Maybe it's really high level at the candidate selection stage, but it's going to drive your entire product development until you get approved. So you
And
have
so
to do
there
that.
is a, and so but the regulatory strategy is global.
If you decide just to launch in Costa Rica, your TPP is just for the Costa Rican market.
No, I know, but I'm saying like theoretically speaking, if you were, I'm more questioning like the alignment between FDA and let's say EMA or other countries. You wouldn't write a TPP for every country.
If your goal, your strategy for this product is global launch, then you will need to know the sync or the dissonance between the different requirements. And then you can say, okay, I need to map out what I need to show and demonstrate clinically for the FDA approval, EMEA approval, or Chinese FDA. And then you do a gap analysis. That doesn't go in a TPP necessarily, but you have to do a gap analysis to understand the requirements and to say, can I design a clinical trial and can I set up manufacturing in such a way that it meets all of the criteria for all the three markets that I have selected? If you plan,
What does that gap assessment look like?
okay, so it's tangentially related to TPP, but it doesn't go on a TPP. Yeah, so regulatory strategy is a separate document effectively. And so you will have to know before you do a regulatory strategy what are the geographies that you want to launch in, and then what are the regulatory bodies that are going to be regulating it, and what are they requiring for an approval in that indication, in that modality. So then you will find the documents, guidance documents from each of those regulatory bodies and put them side by side and say, according to the efficacy, according to the clinical plan, according to manufacturing, impurity levels, reporting, all of the criteria on clinical, quality, commercial, everything, you will create this very non-sexy table and then say, okay, how can I meet the requirements? These guys want to see 2% impurities. These guys are 2.5. So what is the most stringent criteria for me? Of course, in my TPP, then I will accept 2% and not 2.5% impurity levels. So you guide where you want to go and what their ICH is requiring. That's going to be put in your TPP. That in the basically drug formulation section is going to have, this is my purity expectation for the target profile. This is my stability. This is my potency, because you're finding the tightest, the most stringent chain in all of those three markets, those regulatory bodies. But if you just go into Costa Rica, you just build a TPP for Costa Rica and you don't worry about that.
Yeah. So in other words, your TPP will outline which markets you're going to go in and maybe a little bit of detail around which one you may launch in first or second and so on.
Yeah.
But in terms of the regulatory strategy, that goes into a regulatory strategy document. And the further down the line element is a regulatory bracketing where you're saying, out of the, let's say, 3 or 5 countries or whatever you plan on going in, here are the technical requirements for each, such that let's just make sure that we go as high as possible and satisfy all of them at once.
Yeah.
Let's dive into the sections themselves. So we talked about modality and target indication. What else?
So that's number one.
Oh, that's one section.
Yeah, basically target indication is one section.
And it
What
includes
are
modality.
It's a given. So in the modality is going to go basically in the drug formulation section, which is section 6. So we'll get there. I know that as a quality person, you're gravitating. That's going to come. So for
Yeah,
biologists on this call, who are the patients? Why are we building this product in the first place? So that has to
sure.
be in the first one, which is target indication. You define that. And then in the second section, you tell them the mechanism of action. You're saying, I'm developing an inhibitor of this particular kinase, and it has these biomarkers are activated afterwards. That's going to tell you what is the mechanism and what to expect to see in terms of the biology and the clinical output. It doesn't have to be a hefty section, but it has to outline what is the way that this drug works. Number 3 is a very important one, which are the efficacy benchmarks. You have to understand what you're competing against, and you need to put in how do your preclinical efficacy studies need to look like? What are you trying to see? 80% reduction in a particular biomarker or 70% reduction in the size of the tumor. There has to be a very specific efficacy threshold, which if you can't meet in the preclinical stage, you better kill the program altogether. And another area in the efficacy benchmarking, which is section number 3, has to be clinical efficacy expectation. You need to know from section 1 who are you going to be target patients. Then you now have to know how your clinical trial design is going to look like and what you're going to be measuring as a primary and secondary endpoint. You will know those biomarkers. So then you can say in the section 3, which is the efficacy benchmarking, in the clinic, I'm expecting in phase 2 and in phase 3 trial, this particular percentage of my biomarker reduction. You have to be very specific, right? That's what goes in a target product profile. And you can only do that with precision if you know what good looks like. And that's where we select a competitive compound in this section. We say we're going to be competing with standard of care. This does not mean that you're going to have clinical trial design that's going to at the same time compare standard of care or your competitor with yours side by side for superiority reasons. But that just means that you have observed that your competitor has done a reduction in 30% of this biomarker in their trial. So you're going to say, for me to compete, for those physicians to prescribe my stuff, we need to do at least 35% reduction. So that's what you put as your target efficacy threshold. So you have to know what that good looks like. You have to study your competition and then say, I'm going to be competing against this. This is going to be in my label. That's how my marketing is going to be marketing this product down the road.
Yeah. In on the device side, we have this, and I'm drawing a parallel here. We have this concept of state of the art in terms of risk management.
Mm.
And it's said in basically every training under the sun that when people say state of the art, they think this is a state of the art facility, which means it's the best facility, right?
Mm.
Whereas instead of thinking it like state of the art, as in this is the current state of the art,
Mm-hmm.
meaning what you were saying, if you're entering a market for something that already has a quote unquote solution, then you have to at least demonstrate that you're at or better than that.
That's it.
Yeah.
That's it. That goes into TPP here.
Yeah.
Yeah, that's exactly it. So we need to know that even today, before it even hit the patients in the clinic, because we have the preclinical and the clinical efficacy thresholds, if we can't demonstrate preclinically that we can compete, then the recommendation should be to either kill the program or find a different indication, where it's not usually the case. People don't do TPP early enough. They don't know what good looks like in the preclinical. They push it in the clinic, and then it fails in the clinic. If we were more stringent about TPPs and following preclinical efficacy benchmarking, we would be pushing fewer low quality assets into the clinic. And we would be giving the QA and the other groups that are now taking over in the relay game higher quality assets to begin with.
So
That's
the problem, on you go, sorry.
no, go ahead.
I was going to say the problem with not setting expectations is you can have type 1, type 2 error, right? In other words, that if you see an issue in a clinical study, it could be that you overshot or undershot in terms of what you were hoping to see. And so is it all, because you talked about killing the program, is it always the case that a undefined or poorly defined TPP, you get unfavorable or questionable results means kill or find a new indication? Or does it ever mean go back and actually do what you should have done in the TPP and just run it again under the appropriate? Does the question make sense?
I think it does, and that's gonna come in the section 9 of the
Okay.
TPP, which is the risk assessment. What are the
Okay.
risks to the program preclinically and clinically and manufacturing? And how
Okay.
can we mitigate it today? So sometimes you may hit your target product profile expectation target preclinically, but it's just the nature of the animal model that doesn't translate well into the clinic. And there's nothing you can do, right? In that particular animal model, we did hit what we're supposed to do, but it's this type of disease that doesn't translate well into humans. And then you're now left to wait it out in the humans. And that's another challenge that you put it in the risk assessment and mitigations to just monitor, and then you do monitor it. So it
Okay.
delays. And that's why we in the drug development world and come from the investing side, if you don't have good phase 2 data, then you don't have anything. And until you have phase 2 data, you don't really have proof of concept. So until you have completed a well-designed phase 2 study, you will not know if it's efficacious and safe. Everything before phase 2 data readout is a hypothesis. You're just making your best assumption as you can, if that makes
Okay.
sense.
It does. No, it makes complete sense because in phase 1, you're only seeing does it cause harm such that executing a phase 2 is not worth it?
Yeah,
It's not even worth testing.
yeah,
And then phase 2, you're actually seeing does it have the intended result that we
yes,
were hoping for?
yeah.
And I like the everything before phase 2 is a hypothesis. Now, so that's 3
That's 3. 4
efficacy
is safety and
benchmarks.
tox, which relates to this conversation. But we mentioned the FDA guidances for modalities and for indications. We do this section, you read both guidances and you say, okay, the FDA really wants to see non-human primates or dogs or rodents, or these are the species that they want to see. And this is how we're thinking about the safety in animals. And then this is my hypothesis for what I'm expecting to see in the clinic. Is this going to have, based on bioavailability, if a half life is really long, am I expecting some liver tox? Am I expecting some CNS impact, ocular tox? What organs am I anticipating based on my mechanism of action to be of concern? And that's where I notify my quality people. I park it in the risks, right? And then I put it in the section of a TPP for safety and tox. What does good look like? Any adverse effects that I'm expecting? Am I expecting particular organs to be affected? And how does this look like? How do I mitigate it? So that's a very important section. And it is related to the formulation and the route of administration. And we'll talk in section 6. Section 5 of the
But
TP. Oh,
before you move on, for safety and tox, you talked about some of the things like adverse effects. You talked about are you anticipating certain, basically what I heard from you is looking at potential adverse effects, but also looking at the way the potential drug is set up, looking at the human body as a system, what
mhm.
subsystems are likely the most where may we see issues? Is this going to have like potential liver response or is
Yeah.
it going to have potential kidney response and so on? But I guess in my head, tox is a standard thing that you need to make sure that that there's a toxicology evaluation that has
Yeah.
been done, and that includes a number of standard tests. No?
Yeah,
And
yeah,
so
it does.
I think the what I was just describing is the safety part, I think, and then is tox that list of standard tests.
Yes, yeah, that's it. Yeah, the FDA is requiring certain things for the pre for IND talks, and that's
Yeah.
what comes from these guidance documents. Which species do you need to do? How long do you need to do? 28 days chronic tox, reproductive tox. It's very standard. But in a TPP, we need to say what is good like, what look like, what thresholds of data are we expecting? So yeah, the
Yeah.
FDA regulates what you need to do. TPP tells us if we get a result that's above a certain threshold, we have a toxic compound. If we hit the ocular and the liver together in addition to muscle, we have a toxic compound. So that's where the TPP comes in because you're anticipating what data in the future you're expecting to get. And if you get something that's outside of that bound, you need to go back to the drawing board. That's why the TPP is an evergreen document. As you're getting more information, you're adjusting your TPP with all the levers.
In other words, I guess what I'm hearing from you is it like the all of the stuff that you would do as pre-IND talks. Yeah, it's important, but it's table stakes. You have to do that anyways. The important thinking part of this section is
Yeah.
okay, but specifically to this drug, what are the potential safety and tox issues that could come
Yes.
up outside of all the stuff that you were going to have to do anyways?
Yeah, 100%.
Yeah, yeah.
And especially
Okay.
in the clinic, you have to think forward and you can, that's where you benchmark it against your competitor. Hey, in the clinic, they had severe adverse effects. They had a black box warning. They had liver tox. We're going to do better than them. In the clinic, I'm expecting to have fewer side effects, to have better tox profile. So that's what you're doing. You're setting the thresholds of what good looks like. And once you meet it, then you go back and say, yeah, we met it, or we didn't meet it, we can't compete. What do we do now? So there's a section in the TPP. It's the last section, but it's my favorite section, which is go, no go criteria. Because everything is a guessing and then seeing if we meet the guess of what good looks like, what do we as program developers, as drug developers, say is a killable event? And what do we do to mitigate in the interim? So go no go criteria. It's a very soul searching and very emotional section to write where we say, if we get this result, we're killing the darn program.
Sure.
And you have to do it early on, and not too many groups do that. And they keep on developing shitty stuff, pardon my French, because they
Okay.
don't have the guts to include go no go criteria in the TPP and revise it on and
Yeah, and
actually chill
I'm
it
sure.
if they don't meet it.
Yeah, I'm sure that's particularly hard to do with the academic spinouts where
Yeah,
there's a lot
yeah.
of. Okay, so four is safety and tox. Five.
5 is PKPD. So you need to
Oh,
know how is this distributed in the body? How long does it sit in the body? Does it go where it needs to go? All of the fun stuff of ADME. So it's pretty structured, but that kind of also gives you the understanding of the drug index, therapeutic index, and it will drive into the dosing. So what is the dosing that you need to hit preclinically and then in the clinic? So
I'm looking. I looked up the term ADME because I hadn't heard it before. So it's absorption, distribution, metabolism, excretion. In other
yeah,
words,
that's
kind of
a
like
preclinical
what you.
term. Yeah, it's
Yeah,
a very not non-clinical term, and it's pretty standard. You do it in vitro, and then you do it in vivo, and you continue to follow it in the clinic. And you have to know where once the therapeutic goes into your body, where does it end up? How is it eliminated? And how long does it stay? And does it do what it's supposed to do?
and
Yeah.
I'm assuming that this is. I'm assuming this becomes. This section is particularly important for like long acting drugs.
Oh yeah. Yeah. And
I'm sure it's
short
important for
acting.
all of them, but particularly, I was going to say, I'm sure it's for all of them. But if you're trying to make a claim, okay, we're just going back to the section three um, competitive compounds. If you're saying we're going to go from this is a 1 month or daily, And
Mhm.
now with 6 months, that section becomes increasingly important, I suspect.
Oh, yeah. Yeah. Oh, want me to give you another example of the PKPD importance? So
Mm.
maybe we're finding out that our therapeutic is modified in the body and it has metabolites. As it's being processed, it creates certain metabolites. And maybe those metabolites are then toxic, right? So then what do we do? So knowing what happens to the drug in the body and how it impacts the efficacy and safety is very important. And so if it is toxic, what do we do? Do we make a different version of the same drug that doesn't have that metabolite, right? We have to know that because again, tox efficacy, if tox is too high, then we can dose less. If you dose less of a drug, you have lower efficacy. If you don't hit the target, you don't meet the clinical endpoint, you're dead in the water. It's all interconnected. And all of this is so important to think a fully integrated way. And TPP allows you to start connecting the dots and how it all connects.
Yeah, no, 100%.
Yeah.
Um, I
So that
think
was five.
that was five. Six.
Six is the drug formulation. So that's the QMS implication. So what is the formulation? Is what is the route of administration? What is the frequency of dosing? What am I thinking about the impurities? What is my stability? All of that fun stuff is now in the drug formulation section. And that one then turns into critical quality attributes. It goes into like specifications development, and it's one of the very first sections that is turned into quality documents, compliance stuff. You have to know it. But that's where the meat of how do we make the product and what does good look like comes into place.
You talked about release testing earlier.
Mhm.
That goes in this section.
Yeah, that would go in this
Yeah.
section. Mhm.
Okay. So seven.
Seven is the regulatory strategy, and we touched on it. Okay, what's the path? Are we doing just the FDA? Are we doing EMEA? Is this new drug application? Is this 505(b)(2)? Are we doing accelerated approvals? Are we do breakthrough therapies? Is this going to be orphan drug designation? So all of that fun stuff goes into regulatory strategy. And the further along in asset development you polish that up, and that can become an individual document. So all the sections in the TPP are now there to allow fully integrated planning, but each of these sections will then become a separate document. And then each of those will then become additional separate documents, which the evolution of quality or the QMS stems from a TP.
You talked about 505(b)(2), and I'm not a regulatory guy. I'm a quality guy.
Oh,
I'm
sure.
not I don't believe I've ever worked on one that's a 505(b)(2) because it's not exactly an NDA as NDA normally is, and it's not exactly an ANDA. And so, can you describe? I remember looking into this recently. It's funny that you say that. And so, maybe can you, if you can, I don't
Yeah.
know if it's your wheelhouse, but if you can talk about what is the intent of that business case, when and why would you use it?
Oh, yeah. 505(b)(2)s are in vogue today. They go in and out of vogue because it's basically repurposing an FDA approved drug for a different indication. So if you're doing a
Mm.
505(b)(2) regulatory pathway, you have demonstrated to the FDA already the safety to the extent the FDA has.
So you own the drug.
So yeah, you would somehow get rights to use that drug. It's either a generic, right? So then there's no composition of matter IP protection. So you can file IP for just method of use for this other indication. So if you keep, so let's say a drug was approved for one indication and it was IV administered with daily dosing. I'm making it up, but it was for CNS indication. And now you're using it for, I don't know, preeclampsia or some other indication.
Completely different.
It's completely different. So there are certain data that you can leverage when applying for an approval with an FDA. You may not need to do any preclinical studies because the FDA had seen it and approved a certain kind of dosing and the frequency of dosing for this under indication. Then you do this gap analysis, regulatory gap analysis that we talked about, and say, okay, maybe I just go directly into phase 2 clinical trials. So I'm skipping 7 years of all this preclinical and phase 1 work because the FDA knows that this is safe. And I'm just using it for different indication. I'm doing a phase 2 trial with these endpoints. Everybody's happy, right? So that goes into your TPP.
Yeah,
But
yeah.
if you are taking an FDA approved drug that was IV approved for a CNS indication, daily dosing, and you're reformulating into an oral and using 10 times the dose, I'm making it up, and it's for GI tract, the FDA is going to be like, okay, now the ADME is going to be different. PKPD is going to be different. It's going to be absorbed differently. And now, because it's in the GI, I'm seeing liver tox and maybe impurities are different. Dude, you have to go back and redo some preclinical studies, and I might make you do a phase 1 because now it's a different starting dose, right? So you think you're
Sure.
saving time. So there, and that is driven by the regulatory strategy. So all of that you want to know on day 1 in your TPP. Are you a 505(b)(2)? What do you need to do in addition? What not? Are you a straight up NDA with traditional preclinical phase 1, phase 2, phase 3? What do you need to do? All of that you need to know today.
Understood. And in other words, if it's a generic, then you just need to make sure you're either in line with the, say, existing dosing pattern modality. If not, then
Mhm.
you may go back to the drawing board.
Yeah.
If you own the drug and you're going for a different indication, you could potentially use 505(b)(2).
Yeah.
Or if there's some crossover where a drug company has requests rights or
Yeah.
right of reference to existing data, then they can do it. Okay. So that's
Exactly.
section 7.
That's section 7. Just to conclude this thought on 505(b)(2)s, a lot of
Yeah, go.
companies fall in a trap thinking the FDA has approved the drug. And even though I'm changing the formulation and the dosing and the indication, because it was approved, I go straight into phase 2. No, you have to
Mm.
do the gap analysis because the FDA may ask you to do additional safety studies. And so that's why I'm mentioning it here. Don't just
But
assume.
and I'm not a clinical person, so I'm just assuming though. Even if, like, in my experience, like if you're going straight into a phase 2, you'd still have to do some kind of IND submission, right?
Yeah,
Yeah, yeah. And so they may stop you in your tracks
yeah,
if
you're
you
going to be
say
put
this
on
is a
clinical
pre-IND.
hold. Exactly.
Yeah, yeah.
And nobody
Okay.
wants that.
Uh, So the problem is if you've set up everything to go straight into phase 2, then you're back to the drawing board
Yeah, the
if you
joke is
haven't
on
like
you.
actually
So you
yeah,
have
yeah.
to do a TPP to plan it all out. So that was section 7.
7.
Section 8 is differentiation. How are we competing in the market? How are you going to sell this? What's your secret sauce? Are we competing on efficacy? Are we competing on safety? Are we competing on just the ease of use, maybe fewer injections? Are we competing, or is it going to be cheaper? So we need to know that today, and that's how we're developing the product. 9 is risk assessment. We talked about that. What are the risks that we're aware of today? Is it the regulatory risk? Is it the manufacturing risk? Is it the safety talks? Is it the API availability? Is it like super hard to make? So there are different risks. You put the list of risks, mitigation strategies, and unfortunately, the list will grow in each version of your TPP. That can actually be the longest section. The risks
Really?
will just keep growing. You may be retiring a few, but a good product developer will beef up this section, and that goes into the quality organization. That's copy and paste. All of that your quality folks need to inherit. So that's a very important one.
Uh-huh. And then in terms of the mitigate, because you mentioned it a couple times, like that the TPP might be a couple pages long. It may be maybe a few pages, a handful of pages long, let's say.
Yeah.
How detailed do you have to go in terms of the mitigation strategies, or is that something that's handled in later stages?
Mhm. Preclinically, usually it's not regulated, so it's free flowing. Everybody has their own format. Once you hit the clinic, you have an IND open, and then it becomes way more formal, and then it becomes complete new set of documents. So a TPP will just have a section that says risk assessment, and then see additional documents for more detail and clarity, right? As long as your organization has ways to track, it doesn't have to be captured under this particular document, but it has to reference other documents. And yeah,
And then lastly, and your favorite is the go,
the
no
go,
go.
no go. Yes. And yeah, so everybody has their own favorite format. Obviously, the last two,
Is that
not,
actually, I mean, I know go, no go is informal terminology, but is that actually what it's called?
yeah. I mean, for
Like
your
you
own
will go
purpose,
into
it's
the
an
document,
internal
it says go,
document.
no.
It's an
Yeah,
internal
yeah.
document. So yeah, we call it go, no go.
Okay. And the section.
Yeah. And then this section again, it's very soul crushing. And there has to be a discipline behind it and saying, okay, under which circumstances do we go back and modify? And then under which circumstances do we kill? Because we don't know at this point. We're just hoping. Everything's a promise, a hope, and a hypothesis until we have phase two data. So anything goes. So anytime before phase two or even with phase two data, what does phase two need to tell us before we go into phase three, right? And it has to, a good no go, a go no go section also has whose decision is this to make, and then how is this approved at the corporate level in a sense, because it can't just be a scientist deciding, or it can't be the CEO, right? There has to be a racy model attached to who makes this decision and how deep it goes, the responsibility of that. But it's a very critical section of a TPP.
Let's move on. One of the questions I had was, what stakeholders do you think need to be involved in the creation and then refinement of a TPP, and why? And then in my head, it's my way of working, but I personally believe that there should be somebody who's leading an activity. Let's use the example of risk management for a second.
Mhm.
One of the companies that I worked at in the past had a really good risk management process where, yeah, for risk management, collaboration is really important, but they had a role called risk management coordinator. And their responsibility would be to herd the cats, and they would be driving that
Mhm.
activity. Who owns the TPP? Is it marketing? Is it regulatory? Is it
No,
nobody?
no. So it sits with the chief scientific officer. So it's an R&D document. It drives product development, but it can't be completed without full integration of all the functions because it does have, as we discussed, the scientific section. It has clinical, so you have to talk to the clinical development team. You have to talk to QA. You have to talk to regulatory people. You have to talk to manufacturing, and you have to talk to commercial. So R&D owns it. A chief scientific officer effectively is accountable for it, but the responsibility is for the R&D organization to seek input from other groups for the most successful TPP. So I would put it that way. It's not qualities by no means. This is an integral, strategic, and tactical document that drives product development, which is in the R&D hands.
And then the go, no go final decision is typically taken by.
And so that every organization has different decision making kind of structures. So that's why it's important for the go no go to actually specify for these types of decisions, it's in R&D's hands. Manufacturing has a call, regulatory has a call, CEO, right, corporate. So there are certain levels of what happens before we kill it.
And so based on your experience, let's say, what is a good go, no go model? I know you just spoke to it in the abstract, but I'm trying to understand. You get a readout,
Yeah.
right? And then you need to make a decision. It's either perfect or there's some things that you need to deal with.
Yeah. So
And again,
usually,
I'm asking these questions because there are cases, like you mentioned, where the TPP is not very long. So you're not going to have some detailed decision-making matrix with
yeah,
if-then statements that deal with every situation under the
yeah,
sun.
yeah, that's a fair question. So what we didn't talk about in the TPP, preferred format is to have it in a table format. So you will have 10 rows for each of these 10 sections that we talked about, which is target indication, mechanism of action, efficacy threshold. I'm looking at my notes, safety and tox, PKPD, drug formulation, regulatory strategy, differentiation, risk assessment, and go no go. And each of these will have 3 columns. The column all the way to the right will be what do our competitors do? Stake in the ground across all of these. The middle column will be what is our target product profile? The best case scenario on efficacy this, best case scenario on tox this, best case scenario XYZ. But the very first column is going to be minimal product requirements. And you guys have heard that in quality. What's the minimal stuff that we need to demonstrate to swim in this pond? And if
Yeah.
anything is below this minimum, we as an organization need to decide, do we still continue to do that or not? And that usually translates into go no go. So sometimes folks copy that. What's the minimal stuff? Puts that in a go no and say, if we have impurity levels that we can't reduce below 2.5%, we kill it. If we have these certain safety events or liver tox issues below this, we kill it. For efficacy, if we can reduce the biomarker by 55% versus the target 80%, we kill it. It's the minimal stuff that we have to reach, and then you put that in a go no go.
What are there to the I understand what you're saying. You're saying that it's maybe not as abstract as it is in my head because in each section you're outlining the minimum criteria. So like the go no go is going to be a summary of all of the basically the MVP section for each of those. It's just
Mhm.
going to compile them. But how do you handle or do you see are there typically a lot of disagreements when it comes to this? How do you handle who holds the D in terms of decision making within a section? Because I don't know a single maybe marketing person or commercial person who doesn't have some scientific background.
Mhm.
And vice versa, particularly the people who are in a, say, chief scientific officer role, a lot of them have business backgrounds as well. So it's not like the stakeholders in a science space are like 100% science, 0% business. Commercial is never 100% commercial, 0% science. Same thing with quality. And so how do you handle disagreements when it comes to TPP?
For a good TPP, we follow a RACI model. We used to follow a RACI model when I was at Baxter, and I know you're also Baxter. Who's responsible, who's accountable, who's consulted, and who's informed. And
Mhm.
so the section, so the TPP is usually again owned by R&D, by the CSO, and they're accountable for it. Then they have groups that they're responsible for writing certain sections, but not decision making. And then there's groups that are just consulted. And so there can't be too many cooks in the kitchen, but a TPP writing process plus a regular TPP review process, which is usually done on a quarterly basis, brings all these stakeholders in the same meeting room at the same time where we are reporting out on the data that came in as we're refining it and everybody chimes in. So the folks that are consulted or responsible are their voices are heard, and then the function that is accountable for TPP then makes changes and drives what do we do now? So it is a team effort, and a TPP document by default unites all of the stakeholders early on, keeps them talking, and keeps everything rolling smoothly so that there are no surprises.
You talked about the sort of psychology related to the importance of a TPP transitioning from discovery into development before.
Mhm.
I hadn't really heard or thought of it On the device side, we have a similar thing where we say if you've defined the user requirements or if you've written out your design and development plan, you are now outside of the concept sandbox and now you're into the development phase. Can you talk a little bit more about that? Is it like formal that once TPP is approved, you're not in discovery?
Yes and
Is that
no. Yes.
I guess what I'm asking in other words is like, is that an actual thing or is it something that is like colloquially known, but is it
Yeah.
really a formal?
In the preclinical world, the moment where when we qualify a clinical candidate, that's when we go from effectively, we start development officially. And a lot of companies then design a TPP at that stage because they're going to be meeting with the FDA for the pre-IND meeting. That's conceptually and psychologically a milestone that when you know, okay, this is what we're building, this is our candidate. And pre that event, sometimes companies have a TPP, sometimes they don't. They should, but they don't because it's going to help them find that candidate and select that candidate. So in preclinical, I will just say it this way until you start IND enabling talks, the oversight of the FDA in the preclinical development is minimal. And there's a lot of Wild West practices and it's not very structured and it's very kind of freestyle.
But
Groups,
it doesn't need to be, right?
and so that's what I'm going to say. It doesn't need to be. Groups that apply this mindset of being focused and organized and are using these later stage concepts of quality assurance that start a TPP early on are going to have an easier time qualifying that candidate and selecting that candidate and going in front of the FDA sooner. And they're going to be more competitive. So, long story short, the sooner you start with the TPP, the sooner you start actually thinking about the quality and the documentation and report everything, the sooner you integrate all of these functions to start taking accountability for the program early on and have their voice heard, the more successful the program will be. But it's not required, unfortunately, and that's hurting a lot of programs.
But the kind of going back to the transition from discovery into development is that's not yes
Not a
in
hard line. No,
yeah,
that's why.
but broadly speaking, it is this breath of or you release a breath, but now you're like, all right, we're really doing this.
Yeah, yeah. We're really doing this. And a lot of people are just in that limbo and they're like, maybe I do another mechanism of action study. Maybe I still want to understand how things work better and whatnot, as opposed to thinking, what's my efficacy? What's my safety? Can I make this? Can I be competitive? How would this look in the clinic? So as soon as you put down a TPP, even a one pager, you mentally start asking the right questions. Can I compete? Is this a product? How would the label look like? And everything then drives how you spend your time and how you spend your capital and how you focus your teams differently.
Okay. As we move to close, I did want to talk about combination products. And combination products are often looked at as in the generic space, they're a differentiation. In the kind of new drug entity, they're looked at as a delivery mechanism. But I really can't think, I can only think of one case where I've seen the delivery be tied into a, say, phase one, where the delivery system is an important part of how the drug gets into the patient. And
Mhm.
it wouldn't be able to get it. Basically, it goes into, I think, the sort of modality or the route of administration.
Mhm.
But in most cases, the whether it's a pre-filled syringe or an inject or a auto injector or whatever, that usually doesn't get introduced until, say, phase two or phase three.
Mhm.
Have you dealt with delivery systems like that? And is there any tie into TPP?
Oh, 100%. Yeah, we need to understand. So again, the TPP encompasses the product as it's going to be sold once it's approved. So all the
Oh,
components of what is going to be sold and approved have to be a part of a TPP because the integration of the development is tied. So depending on our TPP, what's going to be the dosing? Is it going to be 2 mils or is it going to be 1 mil? That's going to drive the size of the syringe and the gauge of the needle, right? And the labeling and all of that is tied to the what we're delivering. And the sooner the groups start talking, the more efficient the product development is going to be and the better product is going to be developed. So absolutely.
Completely understand. Where can people find you, Marta?
Oh, great question. I'm on LinkedIn, so Marta New on LinkedIn, and I'm at Radius Research, MEW at radiusresearch.com if they want to email me.
And can you talk about what you do?
Yeah, these days I run a CRO, drug development CRO, and I do a little bit of selling, but my team effectively helps lean companies with planning and then execution and the transition from preclinical into clinical stage development of therapeutics. We come from pharma and venture, and so we not only integrate all of these activities and planning across different functions, but we also top it off with what does pharma want to see when you're ready to sell, and what do investors want to see in a TPP today and down the road so you can get that next check. So that's our secret sauce, and it's a lot of fun.
Awesome. Thanks for coming on again, Marta. And as I mentioned earlier, I would love to connect with you guys. So please use the Calendly link to schedule, and we'll see you guys next time. Thanks again,
Thank you.
Marta.
Thanks for having me.